So good afternoon to everyone here present. I thought the tea and the coffee was superb as usual. Now we're starting session number 7, and it's a plenary session with Professor Klaus van Preisner be presenting on his topic at hand. And we'll be having Dr. Banerjee as the chair. So a small introduction for Dr. Banerjee. It's actually an immense pleasure to actually call out for Dr. Banerjee for the chair, because not only has he helped organize this whole event, he's actually been a wonderful and a very great guider and tutor and a teacher for all of us here. Has helped many, many students actually in SSR. So Dr. Indrajit Banerjee is actually a professor in SSR College Medical Department and for pharmacology. He has a passion for teaching and has actively been involved in teaching and a learning process for pharmacology. He has an experience teaching for 15 years and has research interests in clinical pharmacology, epidemiology, systematic reviews, and meta-analysis, alongside with the psychopharmacological drug utilization studies, and medical education and COVID-19. He has won a prestigious CEA prize in neuroscience in 2013 from Kerala, India, and Best Paper Award for the research effect of energy drink consumption on health and medical students. First study report from Nepal at the International Conference of Research and Methodology and Scientific Research in 2013. He has published numerous research papers and various index journals, 6 books and 4 book chapters to his credit. He has presented more than 150 research papers at various international conferences, and recently he was nominated as a Keystone Speaker in the 2nd International Achagra Medical Nursing and Health Science Congress, Turkey. Can you please give a big round of applause to Dr. Banerjee?
Now, I would like to welcome our speaker, Professor Klaus Preisner, who studied biochemistry in Cologne, Germany, and there he received his PhD in biochemistry in 1978. He was a postdoctoral fellow at the Department of Immunology at the Scripps Clinic and Research Foundation, Jola CA, from 1980 to 1983. To work on regulatory pathways of the complement system. Thereafter, he became principal group leader at the Clinic Research Unit for Blood Coagulation and Thrombosis of the Max Planck Society in Giessen from 1983 to 1997, followed by the position as associate professor in 1987 at the Justus-Liebig-University in Giessen. He served as the editor-in-chief from 2003 to 2010 for thrombosis and hemostasis, and is editorial board member of several biomedical journals. He is author and co-author of more than 360 peer-reviewed publications, including cooperative projects with several international groups in the field of cardiovascular medicine. Please give a round of applause for Professor Preisner.
Thank you very much for this nice introduction, and welcome to the final talk, in which I will wrap up some of the cardiovascular research we are interested in for the last 15 to 20 years. I'm really pleased that I'm facing a full house, a full lecture hall at this very last talk, which is not necessarily the case at regular conferences. But I think this is a very special one. And personally, I could extend such a conference or this conference for another 2 days. But because it's such a warm scientific spirit we have endeavored here, and I think I can speak on behalf of all the external or internal delegates. Now, what I would like to do is to present to you a potpourri of aspects in the sort of crosstalk between innate immunity and cardiovascular diseases. On the one hand, some original results from our group or from collaborative work, but also some general aspects that are devoted, as you will see in a minute, to extracellular nucleic acids. You have heard some spots here and there. And the first mention I will do is that not all the work was done solely in our laboratory, but in many instances in very effective collaborations. For instance, with Derek Hausenloy in Singapore, with my principal investigator Sylvia Fischer in Giessen, with Evine Lang in Vienna in the Medical University, and most of all, I would say, with Belle Engelmann and Elisabeth Steindl in Munich. So this is, for me, the easiest slide, and also placed at the very beginning, not to forget to mention all these nice collaborators. Now, the definition of innate immunity implies that this very early response of our body towards cellular stress, internal stress, also called steroid inflammation, for instance, by trauma, etc., without the participation of external factors, such as a dog bite, is perceived with the participation of a number of cellular and humoral factors, as you see in the lower part of the slide, but also in response to external factors, microbial, bacterial, and viral factors, that all then are defined on the one side, so-called danger-associated molecules, if they are based on the internal response, or pathogen-related or associated molecular patterns, if this has been an external inducement. And these PAMs or DAMs are recognized by a number of receptor sites. We heard about toll-like receptor 7. That's just one example. The response then continues into immune cell recruitment, inflammatory reaction to fight this cellular stress, and in some cases, adaptive immunity using B and T cells are called upon to further resolve and regenerate this response. Now, how can these DAMPs, these usually intracellular molecules that are released from cells upon various types of cell stress, reach the extracellular space? You see here some of the principal mechanisms, including apoptosis, pyroptosis, peroptosis, and also necroptosis, which is one of the major mechanisms how to throw out, so to speak, certain types of extracellular nucleic acids. And you will see in a minute that another type of liberation of nucleic acids, namely these extracellular traps, these decondensed chromatin structures, DNA histone complexes we heard about, is kind of a new type of release of DAMs. Now, DAMs include molecules from the nucleus, from the cytosol, mitochondria, etc. I don't want to go into detail here. Entail proteins, nucleic acids, even ATP is a GAMP, if you want, and with particular emphasis on nucleic acids, which I will concentrate for the remaining time of the talk. We can distinguish cell-free extracellular DNA, mitochondrial DNA, quantitatively less in comparison, and the already stressed neutrophil extracellular traps. With regard to the self-extracellular RNA that can be released on various types of cell stress, we distinguish the majority of release RNA that is ribosomal RNA. About 80 to 85% is ribosomal extracellular RNA. When I talk about extracellular RNA for the next few minutes, I mean particularly this ribosomal extracellular RNA. But this is not the only type. We also can talk about long non-coding extracellular RNA. Also, some messenger and transfer RNA can be released. And particularly interesting, as we also heard about different types of more than, I think, 1600 of microRNAs. But again, the extracellular RNA I'm talking about is the ribosomal type. In addition, there is another specialty that these types of nucleic acids can be released in free or naked form, if you want, in association or packaged by microvesicles, or in association with proteins or other positively charged molecules. So once these dams have been released from necrotic, immune damage, whatever cell stress tissue, they are initially called alarmins or danger molecules to make the immune system aware of such a cell stress or tissue damage, whatever. They will be recognized by different types of receptors, as we heard, and thereby initiate a cascade of reactions aiming to induce cytokine release, which also can work on further alarming or danger molecule induction, finally resulting in an inflammatory response, hopefully not in a hyper-inflammatory. And as you can imagine, one type of dams, alarmins, is represented by this already mentioned extracellular DNA and extracellular RNA. And there are several ways to counteract intrinsically these alarming response by blockage or neutralization of these ones, also pharmacologically by interfering with the receptor induced mechanisms, as we heard some time ago, or to block the cytokine. And all these different dams work also on different cell types, macrophages, vascular smooth muscle cells, or dendritic cells, in the context of vascular diseases, atherosclerosis, heart attack, etc. And you see a number of these dams have been identified and characterized as not only alarmins, but also as really damaging molecules. But the higher the extent concentration of these alarmins is, the more damage they can induce. And this is, as you will see in a minute, particularly true for extracellular RNA. Still, I want to begin and start with extracellular DNA. In the second part, then hop over to the extracellular RNA. So I already told you that there are different types of extracellular DNA. How are they distributed? could they serve as biomarker? What are their functional properties? How is the disease, the connection to diseases and other antagonistic approaches? Now, this is a typical picture of the already stressed neutrophil extracellular traps called nets on a light microscopical level with a particular stain. And you see these nets derived from activated neutrophils with a DAPI nuclear stain. And although this is so obvious, also for a student experiment, it was only in 2004 that Volker Brinkmann, a colleague in Berlin, discovered these structures. And if you look closer by electron microscopy, and this is a typical picture that Volker Brinkmann took, you see that these in yellow nets are protruded from neutrophils and can catch and kill these red-stained bacteria. So this is an example for an extracellular killing of microbes, as opposed to phagocytosis as an intracellular killing. And if you even go closer, you see these net structures very much ordered along the DNA strands are sitting proteins, histones, but also highly concentrated granular products from neutrophils, these antimicrobial proteins, enzymes, etc. They hook up to these nets, become highly concentrated on that surface, and are able, as I mentioned, to kill these bacteria. If these antimicrobial peptides would have been released into the soluble phase, their concentration would be much too low to be effective as an antimicrobial effect. So what I just indicated was we have phagocytosis as an intracellular killing mechanism and netosis as an extracellular one. And the initiation of netosis is induced by very many stimulators of neutrophils, including shear stress, heat, cytokines, bacterial factors, just name it. And the intracellular chromatin will decondense due to a specific mechanism I will show you in a minute, and then within 4 to 5 minutes you can set the clock. Actually, this decondensed chromatin will be released into the vicinity of the dying apoptotic neutrophil. So agonists, as I indicated, are represented here, and the mechanism schematically is shown here. At the end, you've got these nets, the blue DNA strands, histones, and antimicrobial proteins are associated to kill the bacteria. And this is partially dependent on a NOx, so a reactive oxygen species related mechanism, but there are alternatives as well. And a enzymatic activity based on protein arginine deaminase 4, which means that this enzyme deaminates certain arginine residues on various histones that are responsible for the tight ionic interaction between the positively charged histones and the DNA. And if this tightly associated ionic interaction become loosened or even weakened and lost, the chromatin decondenses and is released. In fact, you will see it in a few minutes that in the absence of PET4, so in a knockout model of PET4, no nets will be formed. So a nice way, perhaps also clinically, to reduce net thrombosis. And lastly, this modified citrulinated histones are also a specific marker, monoclonal antibodies, for instance, to serve as a quantitative measure for NETs, tissues, etc. But there's another aspect that certain bacteria, you see here some streptococci caught by these NETs, that certain bacteria express on their surface DNases. How nice. So they just digest these NETs and can escape, so they are resistant against the process. Keep this in mind for another case with DNA. And due to the action of neutrophils also with other companion cells like platelets via several adhesive interactions, Bernd Engelmann in his lab found that there is an interesting correlation between the formation of such aggregates or cellular conjugates and the formation of NETs. Together with his lab, I think in 2003, these data were sort of collected before NETs were discovered, but what we saw indeed was the same aspect without having a definition for this process. Anyhow, in addition, NETs, existing NETs can furthermore acquire platelets that adhere and further induce thrombus formation and NET association. So these cell aggregates, which is also known for quite some time, are measured for cardiovascular and chronic inflammatory diseases, but people have not had a clue about the mechanism. Now we know what the mechanism is. And I will now turn from the sort of antimicrobial, the defense side of NETs, into another aspect that is indeed related to the platelet neutrophil aspects that I just introduced, also in association with the platelets. Because in both cases, in the upper part, arterial thrombi, as well as in venous thrombi, there are NETs operative also causally to induce thrombus. So each arterial or venous thrombus contains NETs besides fibrin, cellular debris, etc., and so on. And let me show you a few of the data we have collected. Together with Irene Lang in Vienna, we saw in authentic coronary thrombi from patients pretty significant staining of NETs in these thrombi. Blue again are nuclei from neutrophils, monocytes, and other nucleated cells. And if you look closer, there is really a heavy deposit, if you want, of NETs within these thrombi. And this correlates very well with the location of neutrophils. And again, the mechanism how these NETs appear there is due to the platelet, mostly platelet neutrophil interaction, besides some other aggregates. And if you look and compare coronary, venous thrombi, the density of NETs, represented here by the red stain, is quite high. In venous thrombi, there is something, but not that high. And if you produce an ex vivo blood clot from plasma, there is hardly anything visible in terms of NETs. So this is for arterial thrombosis, coronary. What about venous thrombosis? Also together with Bernd Engelmann and other colleagues from Munich, using 2-photon microscopy, one can demonstrate the accumulation of platelets and neutrophils on the intact reservoir in veins. And again, what will be formed are nets. So these are nets indeed. And interestingly now, if one applies DNAs under these situations, you see there is hardly any thrombus weight. There's hardly any thrombus form. And the same is true, as you would expect, in the presence of infused DNAs. There are no nets build and accumulate. So in other words, DNAs appears to be an anti-thrombotic agent, a new anti-thrombotic. And even more surprising for us was that even low molecular weight heparin, here enoxaparin, like DNAs, was able to block net formation. I think there's hardly any doctor on earth who would connect the heparin infusion they do million times wherever in hospitals, would associate it with the inhibition of NETs. Certainly, heparin is a cofactor for the anti-thrombin reaction, yeah? But this is the initiation of the thrombin. So what we think is also happening for the induction of a venous thrombus is the accumulation of such nets on the endothelium. And you will see in a second, this creates a cytotoxic material. Endothelial cells are activated, destroyed, become pro-thrombotic, pro-inflammatory, and that will accumulate into a venous thrombus. And here are the data. So nets are able to induce cytotoxicity in endothelial cells and epithelial cells. We heard from Sean Davidson that histones might be an interesting inducer here. And indeed, this is the case also for nets, because in the presence of antibodies against histone 4, or in polysialic acid, which we had available in the lab, the polyanionic compound that complexes histones also in the presence of nets. You see there is hardly any cytotoxic activity. And finally, nets are associated with a number of vascular diseases. Again, you see in an atherosclerotic model the deposition of lipids here, atherosclerotic plaque, area, etc. In the presence of DNAs, you hardly see any of this deposit. This is here quantitated. And one can, in fact, distinguish three types of nets in such atherosclerotic lesions. So-called intravascular nets, which are mostly fragments of nets that can circulate, lesional nets, and prothrombotic nets. I don't want to go into further detail, but just to give you the kind of detail. And now coming to the sort of final aspect of this part, and you can already expect what is depicted here. There are several ways to inhibit the formation, the production, or the progression of nets. I have given you a few examples with DNA. So, with DNAs as an antagonist or an antithrombotic agent, but also TAT4 is one of those, and there are several other approaches. I don't want to go into detail here, that I think will be developed in the next year. And finally, there's also nets in stroke thrombi, as indicated by a publication some years ago. And again, situated histone 3 is a specific marker, and you see in this stroke thrombi, this thrombus is full of nets. Now we have, together with the renal lung, thought, well, TPA, tissue plasminogen activator, is one of the, or maybe the only thrombolytic agent used for thrombolysis here in situations. If thrombi are full of nets, why not using a combination of DNAs and TPA for dissolution of the? And indeed, these ex vivo thrombi can be lysed much better with a combination of TPA and DNA. So that might be also a new way for neurologists, other people, other clinicians. So just to sum up, there are two major activities of neutrophil extracellular traps. One, to fight infection. You see a bacterium is sitting, happily or not, on a net, and to the right you already recognize this figure in this coronary thrombus, which is full of net. So they participate in vascular thrombosis. So and now come to the second part related to self-extracellular RNA. Again, you have seen what I mentioned, and I'd like to show you a little bit about the distribution, the functional properties, relation to disease, and antagonistic approach. This is a picture of a very early work, also together with them, where we induced in arterial thrombus non-invasively by a chemical method. And within several minutes, this occlusive thrombus is formed and accumulates, and this is the green color, extracellular RNA in a very heavy way. This fluorescent dye just is a complex agent that detects extracellular RNA. In another example, in atherosclerosis, from the timeline, several hours, the first picture, 2 days further on, and 4 days here in the development of atherosclerosis. You also see in green the accumulation of extracellular RNA on this diseased vessel surface. So there must be something going on that is not associated with extracellular RNA as a biomarker, but obviously also with this nucleic acid as a functional entity, a pathophysiological entity. And in fact, the higher the stress, whatever it is, the more extracellular RNA accumulation you will find. One example is here for a hypoxic induction in different tumor cell types without apoptosis, without mechanical damage. They are still intact. The only inducer is hypoxia. You see this massive increase of extracellular RNA. And this is also associated with the deposition of extracellular RNA in authentic specimen from glioblastoma patient. And next to this glioblastoma site of the patient in the white matter, you only see the excess or the extra and also intracellular RNA within the cells. This is the thrombus association you are already familiar with. And another example, now coming to this very much stressed keyword here, ischemia reperfusion injury, where we did a study together with Derek Hausenboy and colleagues. You see first of all, the nature or the identity of ribosomal extracellular RNA which is nothing else composed of the 18 and 28 S derivative component. You will also see if you isolate cellular RNA from any type of cell. And by analyzing in parallel the messenger RNA that is within the specimen, you see that the majority of messenger RNA was derived from SERCA, so a cardiomyocyte-specific gene or protein, indicating that under cardiac ischemia reperfusion injury, cardiomyocytes are the cell that releases the majority of extracellular RNA. And this is just simply to give you an overview about the different functional activities and properties of extracellular RNA, also as a cofactor for different cytokines, for instance, vascular endothelial growth factor, in thrombus formation because it's a cofactor for contact proteins like factor 12. It induces leukocyte adherence in the recruitment into the vessel wall. It can polarize macrophages from the N2-like into the inflammatory N1-like phenotype, and also, as we just have seen, induce cell death in cardiomyocytes. Now, a very simple way to protect cells or to interfere with these damaging properties of extracellular RNA is the application of RNAs, specifically RNase 1. You remember in the DNA chapter, we have the extracellular DNA and the DNase, and similarly it works also for the RNase chapter. And the majority of natural RNase 1 is derived from endothelial cells. So along our complete vascular tree, we do have constitutive expression and release of RNA, homeostatic balance with any type of particularly ribosomal RNA that is around. And as I already indicated, there are very many situations we also described over the years in cardiovascular diseases that are related with the induction of extracellular RNA and the antagonistic function of RNase 1. This is one example that RNase 1 treatment in a APA negative mice, you will see here, can definitely reduce the deposits or the plaques in the neo-ethanol, but not in the medial. Yeah. Just to keep the story short, this is in a table of a few of the examples we studied in these different preclinical models. And just by reading the red keywords, you see RNase works as an antithrombotic, vasoprotective, anti-atherogenic, and so on and so forth. As a real body's own internal vessel and tissue protective agent. So I bet there will be a drug in a few years. But before we really can buy such a drug in pharmacy, we should do something else. Another example, I think. But the one remaining question is, how is the mechanism of tissue damage by extracellular RNA? Obviously, the tolerant receptors, particularly tolerant receptor 7, is not a sort of recognition site. And what we discovered was that exposure of immune and other cells was extracellular RNA increases significantly the induction, the induction of TNF alpha. And TNF alpha is not simply released as an active cytokine, but it needs to be proteolyzed from a membrane-anchored form into the active trimer. And if one uses the protease inhibitor for this process, you see there is also no effect of extracellular RNA operative. And what we think happens is that extracellular RNA mobilizes or induces this protease, which is called tumor necrosis factor alpha converting enzyme, or ADAM17. So extracellular RNA triggers this membrane-bound enzyme that then is able to process the membrane-bound proform of TNF alpha into the active form. And we have many data to show that the appearance of extracellular RNA goes along very tightly with the increase of TNF alpha. If you, even in patients, if you reduce the extracellular RNA, also the level of TNF alpha. And what happens is, what we think happens is that this process here, that extracellular RNA triggers ADAM17 that can mobilize active TNF alpha. And this brings me to the summary of this cooperative work with Derek and other people, how we envision the damaging effect of extracellular RNA in ischemia reperfusion injury. So there's extracellular RNA released massively in the hypoxic area. It then triggers TACE, as we just heard. Pro-10F is converted to the active soluble form, works on the sort of inflammatory, pro-inflammatory TNF alpha receptor, and that all leads to cell death mechanisms without going into detail into cell death. And interestingly, these amounts of TNF and other cytokines then also amplify the whole process because they induce release of further extracellular RNA. And there are two antagonistic ways to reduce this, either by an inhibitor of this TACE enzyme or, as we just heard, by RNase. And in the last two slides, I will give you some information on this mysterious or pretty valuable RNase-1. It's a member of the soluble extracellular RNase A family. It's the most thermostable enzyme of our body. You can cook it, throw it against the wall, survive all these treatments. It is non-toxic for host cells because all cell types contain an absolutely high affinity RNase inhibitor. So as soon as this and other RNases enter the cell, it's immediately inactivated. And as we already heard, it's a vessel and tissue protective enzyme. The only sort of disadvantage is, at least for therapeutic reasons, that its half-life is just 30 minutes. So if you want to use it as a clinical application, I mean, you need tons of it and should infuse it online. So, in addition to its synthesis in these are cultured cells that release it into the supernatant, there is a portion still in association with the cells. So it might be stored in some entities and vesicles. And indeed, this is the case. endothelial cells contain these typical myelopalatid granula, where TPA, von Willebrand factor, IL-8, etc. is stored and concentrated, and the same is true for RNAs. So in cases where you stimulate endothelial cells also in vivo, there is a short-term increase of the enzyme in the circulation. This is an experiment in mice, but we also have done this with students, with medical students. It works. Now, how can we come along with a therapeutic formulation of this enzyme? We tried to hook up this enzyme to albumin in order to increase its half-life, but this has failed and we did not really further pursue this. But in recent studies from Resolve Therapeutics in Miami, they managed to hook up two RNase molecules in exchange of the FAB domains of a typical IgG1 entity. And there are two studies where they showed the safety, the positive effect of a therapeutic agent, etc., in autoimmune disease. And sort of the identity of this fusion protein, it is non-toxic, has no immune reaction, and now the most important aspect, its half-life is 19 days. So if you want to use it as a therapeutic agent, you may inject it maybe 2 or 3 times a month for effective. And here's just the protocol of such a study for Sjogren's syndrome, and showing here the score of mental fatigue in these patients is clearly different between the placebo in blue and the fusion protein RSLD 132. And what we're going to do now, and we have started in several directions, also with collaborative partners, to use this drug in different cardiovascular disease states. And perhaps if we meet next time in Istanbul, and if I'm allowed to speak again, we might have some initial data. So that was it. And thank you very much for your attendance. And, one message for all of us, for any time, keeping glad and thoughts. Okay, that's it.
Thank you, Professor Klaus, for your wonderful talk. Would you like to take some questions?
Sure. No, if you want.
No, we won't take too long. So it's open for question and answer. Anybody? Yeah, it is our medical student from SSR Medical College. Yeah.
Thank you for this speech, sir. It was quite amazing. So my thoughts are on the presentations which were held yesterday. For example, Professor Yellen started off with the D132 pathway, and Professor Sean continued it with, I believe, histone 4 toxicity. So in continuum of that, in mitosis, as you discussed today, it seems as if toll-like receptors are the common culprits for all these 3 pathways. And we know that TLR agonists are used in the treatment, well, at least are being tried in the use to treat shock. Is there any scope for the use of TLR antagonists to achieve the same cardioprotective result?
Well, first, it might be, as we have seen in the other presentation, but whether this has anything to do with an anti-extracellular RNA effect, I'm not sure, because we could not accumulate data to show that this ribosomal extracellular RNA is really recognized by TLR7, by TLR3, TLR8, etc. Other people can do it, but they use very unclean ligand mixtures. They just scratch tissue and then use this as a kind of ligand. But what I should say is that you I've mentioned this, that in association or the association of whatever RNA, extracellular RNA molecule and a positively charged protein, perhaps another DAM, would give a chance of recognition by TLRs, etc. We have shown this for TLR2 agonists. No time to show it all. For instance, for a lipopeptide ligand for TLR2, and the fact is that none of the single ligands, so neither extracellular RNA nor the lipopeptide as a single agent, does any induction of a TLR2, but if you put them together in a synergistic way, there is quite a bit of immune response or induction of TNF, etc., in macrophages and so on. And I think this is one mechanism how to also envision the action of extracellular RNA in other situations.
Thank you. Any more questions from the audience? Yes, please.
Thank you very much. Very interesting. Just one short question. You said that the interaction of platelets with neutrophils causes net formation.
Among other things. Yeah.
But not always. So we, in our system, we do not find any net formation, and so we have this.
You mean in the collateral artery?
Yeah. So we find a lot of PNAs without any net. So any idea about the regulation?
First of all, one should be sure that the cells are neutrophils and not monocytes, because platelets also form aggregates with other blood cells.
Yeah.
And then there might be, dependence on the concentration, on shear stress, on other perhaps human factors that prevent this kind.
Yeah.
I don't know whether Bernd has a clue for that.
Yes, it's I think it would be an oversimplification to say that whenever platelets encounter neutrophils and form complexes, for example, via P-selectin PSGL1 interactions, that neutrophil extracellular traps are being released. For example, you need a specific way of activating the platelets, and you need a specific context. So it's not a general property whenever you have a contact between platelets and neutrophils that nets are being released. For example, we have done it with collagen, type 1 collagen. Yeah.
Wonderful talk. It's I really got a very good education on the extracellular RNA and DNAs. I have one question is, when you use the DNAs or RNAs, you chopped off the the nucleic acids, the RNA or DNAs, but they may become short fragments, right? And my question is, do we expect activation of those toll-like receptors like a 3, 7, 9, 8 in the human case? And the second question is more about the other parts, the non-nucleic acid parts, like the histones. So where are they? So I'm just get a bit confused.
Yeah, regarding the first part, whether extracellular RNA fragments that are small enough maybe to penetrate or to be taken up by cells and then reach the endosomal TLRs might work. Yes and no. You need a trick to perform such experiments with certain agents that are used for penetrating cells, penetrating membranes, etc. Then it might work. On the other hand, these self-extracellular RNA molecules, pieces, etc., are not the same from the structure and from the base structural point of view as foreign ones, bacterial, viral, whatever, that are indeed recognized by TLR7, etc., due to some modifications. And these modifications are more or less the same one that have been identified and then used to be manipulated by the recent Nobel Prize winner Katalin Karikó in order to not recognize messenger RNAs that have been produced by the immune system. And this is not unimportant because if self-extracellular RNA would be recognized in any way by TLRs, then we would have an overflooding, hyperinflammation, autoimmunity anytime. So it's also a structural difference, not only size-wise or with helper molecules, but what really determines all these different factors is still a matter of research.
But for instance, the autoimmune disease patients, right? So they do report the TLR7 or TLR8, they recognize the self RNAs. So that's, I don't know, but maybe there's a sequence specific. But my second question about the leftovers, so the influence of other proteins, once they are released from the cell into the extracellular, space, these become sort of dense instead of, our own part.
Well, there's perhaps even a chain reaction due to the fact that on the vessel wall surface, on endothelial cells, and also on immune cells, there are at least two ectonucleotidases expressed, CD39 and CD73, that might convert ADP into AMP and AMP into adenosine. And we haven't analyzed this in detail, but these might be the end products. And you might recall that adenosine is a kind of cardioprotective agent as well. So the cardioprotection by RNAs might not be just fragmenting the damaging high molecular weight RNA, but also leading to the split products with the help of the other enzyme.
Thank you. Thank you very much.
Thank you. We won't take any more questions. And thank you, Professor Klaus, for your wonderful talk once again and for today's video.
Okay, thank you very much.
Thank you, Professor Presner, for that enlightening talk. We'd like to call upon Professor Banerjee to hand over a certificate to Professor Klaus Presner.
May we please call Dr. Derek Hartnett to give Dr. Indrajit Banerjee his certificate, please. Thank you.
Now we would like to hand you over to our MCs for our closing session. And before we do that, please don't forget to scan our QR code and give us feedback for today's session. Thank you.
Good evening, everyone. It's great to see you all again. As we near the end of the scientific section, I would like to call upon Dr. Seewoosagur from the College-University, Istanbul, and she has a very exciting announcement to make.
Yes, first, I want to thank organizing committee and SSR Medical College for organizing this well-organized, valuable scientific meeting. And I would like to invite you to Istanbul for 8th International Symposium of New Frontiers in Cardiovascular Research. The meeting will be placed in September, but the date, exact date will be confirmed in a few weeks. Istanbul is a beautiful, unique city with its historical landmarks and also unique Bosphorus, enjoyable entertainment life. And also the host of the meeting will be Koç-University, and Koç-University is high-ranking international university, and which has valuable scientists. And so I hope we will meet in Istanbul in 2024 for the next meeting.
Thank you so much, ma'am, for coming up. Ladies and gentlemen, it's with immense joy and admiration that we stand before the young minds that presented their posters today, and along with the young investigators. We have been in awe of their confidence and knowledge. So can we please have a round of applause for all the participants? So it is time for the much awaited results. First, we'll have the prize giving for the poster competition. The second runner-up goes to the team of Yahil, Fatima, and Videha. That's it. Could we please have the team on stage? Okay. Can we please have Professor Peter Ferdinandi on stage to give away the prizes? The first runner-up is the team of Kriti Gupta. We have Kriti Gupta, Ranjit Sharma, Ankita, and Runda. You can take the center stage. And the winner is the team of Belle. It's Ajay Murugan, Belle Murugan, Sitesh, and Jabez. Dr. Ferdinand. Yeah. The title of the poster was Clinical Outcomes of ICDs versus Catheter Ablation. Thank you. Can we please have on stage Professor Malcolm Walker to give the certificates for the best investigator?
The second runner-up is the team of Kriti Gupta. Again, we have Kriti Gupta, Vrunda Jadhav, Prinjim Sharma, and Ankita Trivedi. The title of the session was Prerequisite Screening of Aspiring Athletes to Prevent Disaster. So can you please take? The first runner-up is the team of Isha Gaikwad. It's Isha Gaikwad, Ishita Sharma, Sudiksha Chaturvedi, and Vedanta Sri. The title was Global Trends in CVD Risk Management due to the Smart Wearable Devices. And the winning team is the team of Akshay Sati. We have Akshay Sati, Vedanshi Tanna, Karina Panwar, and Petri Tharuni Reddy. The title was A Systemic Review of PCI and CABG in Chronic Coronary Syndrome. Thank you, sir. You may take your seat.
I would like to take this opportunity to acknowledge our very dear resource persons, Professor Indrajit Banerjee and Professor Dr. Harshal Sabani. The resource personnel conducted a 7-week of intensive course on research methodology from the 15th of July 2023 to the 26th of August 2023 and guided the participants of SSR Medical College through the entire process. The workshop covered introduction to research, formulation of a research question, study designs, oral and poster presentation, topic selection, and other advanced searches. The students successfully completed the workshop, gaining valuable skills for evidence-based medicine and research in the future medical careers. Now, I kindly request Dr. Klaus Priesner to present the resource person with their certificates, Dr. Banerjee and Dr. Sabani, who could not make it today. So can I request Dr. Klaus? Thank you, sir. You guys may take your seats. The local organizing committee has been the crucial link in the successful conduct of the 7th International Symposium of the New Frontiers in the Cardiovascular Research, and we're deeply grateful to each one of them. I kindly request Professor Bon Engelman to honor our committee members with their certificates. If you may kindly come on stage. Firstly, I would like to call Dr. Rama Srivastava. Sir, can you see on the seats? Second, we have Dr. Faraz Ahmed. Dr. Amina Ahmed. and Dr. Priyanka on math. Thank you so much, sir.
Ladies and gentlemen, the last 2 days of this enlightening scientific session has been a journey of discovery and knowledge. We've dwelled into the depths of wisdom and gained profound insights. Now it's time to honor the dedicated core committee members who made it possible. And to express our deepest gratitude and appreciation, we would like to call Professor Carl Wilhelm Vogel to give the core committee their certificates. First, we have Professor A.P. Singh. Next, we have Professor Derek Hossin-Lloyd. Sir, can you take your seat on the stage, please? Professor Hossin-Lloyd. Sir, can you please take your seat? Next, we have Professor Klaus Priesner. Can you take your seat on the stage? Next, we have Professor Indrajit Banerjee. Thank you so much, sir. We're nearing the end of the session, so I would like to request our core committee members to say a few words about their experience over the last 2 days. First, I would like to welcome Professor A.P. Singh for his word.
It was a wonderful experience. Last 2 days were very, very enriching for almost every one of us. And I'm very impressed by our students who have done a wonderful job in poster presentation and participation and their volunteers. They need a big round of applause. And I'm quite thankful to Professor Derek, and Yellen and Professor Klaus. They are very big guiding force for all of us. And I think there will be good collaboration between our students and these topmost researchers in the faculty and in this field. I hope they will take advantage of it in the future as well. Thank you very much.
Professor Hossin-Lloyd, can you please have it?
Hi everyone, and it's such a shame that it's come to an end. I hope everyone has enjoyed the last 2 days of conference that we've had. I'd like to thank the speakers, the audience, and the local speakers. I think it was a really good blend of science from overseas, as well as science here, but also the cardiology practice. Now I need to say some thanks. First of all, I'd like to thank all the organizing committee, the helpers, and the medical students who helped with the organization of this meeting. I must say, out of the Frontiers meeting, this was one of the most organized and well-run meetings, especially with all the AV stuff and everything went very smoothly. So I think we have to do a big thanks to all the students and volunteers who made the meeting possible. And then finally, I'd like to give my heartfelt thanks to Indrajit and Dr. Singh, who made this happen and organized this, of course, together with the team, and who made this happen and have really made this a splendid experience for us visiting. Thank you very much.
Professor Giessen, can you please add you?
Thank you very much. First of all, I can only assist, underline, and express in bold letters what Derek just said. A big, big thank to the local committee, especially to the head of the organizing team. It was really a pleasure and to work together with you also in the weeks and months before. I think if I were to choose another cooperation or another meeting with a cooperation partner, I mean, one of the first selections would be you as a cooperative agent, scientific agent. And you created together with especially the student community here, such a warm scientific atmosphere and spirit. I wish we had such a spirit and atmosphere in certain cases also in institutions in Germany. So my wish is keep your emphasis, your enthusiasm, of course, your dedication for asking more questions to find concrete answers in the medical field for the next years. And I would invite those who might be interesting for a kind of lab rotation or visit in one of the labs in Giessen to contact us and to find out under which conditions we can arrange such a thing. Thank you also for the young people here, all the technical experts, the introductory speeches, fantastic, I must say. And as I said, I could continue for another 2 days in this kind of, but at an end, we make an end, and this is the end. And thanks very much for all the hospitality.
Thank you, Professor, for the encouraging words. Next, we have for the vote of thanks, Professor Banerjee.
Ladies and gentlemen, on the behalf of organizing committee, we'd like to extend our heartfelt gratitude to all of you for your presence and active participation for 2 days of International Symposium. Your support and enthusiasm have truly made this gathering a memorable and successful event. I extend my gratitude to our Honorable Chairman, Sir, providing us an opportunity to organize such an event. I'd like to thank Dr. Singh, Sir, who always supported me from the beginning. Always he said that, go ahead, go ahead, go ahead. Whatever the problems are, go ahead. And this is my third conference that I'm organizing. This is my third in-person international conference. First conference we have organized in 2013 in Nepal. That was by International Epidemiological Association. I was also in the committee, organizing committee. What we have learned, when that time I was working in Manipal, and what I've learned there is that you don't come with problems, you come with solutions. Always we thought from here, from the beginning, that we were working with this team, and I was always trying to find a solution, not the problem, because problems are many, difficulties are many, but we are trying to solve the problem. So I'd like to express my gratitude to the 4 organizing committee, especially Professor Derek Yellen, Professor Derek Counseley, Professor Claus, and all the delegates, overseas members, those who came to Mauritius all the way to visit us. Our special thanks to the Cardiovascular Society of Mauritius and the Cardiac Center, those who have accepted our invitation to collaborate with us. I'd like to extend my appreciation to the local organizing committee, Dr. Faraz, Dr. Amina, Dr. Rama, Dr. Priyanka, and Dr. Harshal. I'd like to thank the management of SSR Medical School. I'd like to mention the special support that we got from the beginning, especially Ms. Kitty and Mr. Jatin for constant encouragement and support. I'd like to thank all the faculty members of SSR Medical School for their support and active participation in the 2 days of symposium. I'd like to thank the Department of Pharmacology, Dr. R.K. Gupta sir, Mr. Amarendra, Dr. Nagpal. Special thanks goes to our non-teaching staff of our department, Mr. Ravi, who has helped me to the plan with the logistics initially that we were doing around 8 months before, and Preeti also from our department. Special thanks goes to all the volunteers. Without that, without their help, this event, I would say that it was very difficult for us. Like I already told that we come up with solution. The solution was you, right? So thank you all of you. A big round of applause for all the volunteers who worked hard for the 2 days. Next, I'd like to thank all the students, those who participated in the poster with full energy. Is the youth with energy that we've seen. And we have seen that up to which level our student can also reach during the presentation. All of you have experienced that during our oral presentation as well as a poster. And then I'd like to thank all the non-teaching staff of this SSR Medical School. It is not possible for me to mention all the names, but if I missed anyone, I'd like to apologize for that. I look forward to host such an event in future also, if opportunity is provided by our principal sir. I'd like to be happy to organize again. Thank you very much.
Thank you, sir. In closing, we want to extend a heartfelt gratitude to the core committee members, the local committee members, the guests, the esteemed college faculty, the enthusiastic students for making this scientific session a resounding success. Your unwavering support and hard work have truly enriched our pursuit of knowledge and innovation. Thank you all for your invaluable contribution. Have a great evening and safe travels.